Host microorganism interactions. These are your objectives for this lecture. All right, so for an introduction, we have some definitions. So colonization is the persistence of organisms in or on a host without causing disease. An example would be Staphylococcus aureus in the nose of a healthy individual. Symbiotic means a close relationship between a host and a microbe, often part of a normal flora. So these organisms live together. They may or may not benefit from each other, but they do not harm each other. Mutualistic is a type of symbiotic relationship where both organisms benefit. So an example of this would be like gut bacteria that help in digestion while receiving nutrients and a habitat to live in. Parasitic, by definition, is a relationship where the microbe benefits while causing harm to the host. So an example would be a tapeworm in a human intestine. Then you have zoonosis. This definition is diseases that are transmitted from animals to humans. So examples would be rabies, Lyme disease, and even avian influenza. Host-resistant factors. So here's a little bit of an immunology review. In your host-resistant factors, remember that your innate immune system or innate response is something you're born with, and this is non-specific. Innate resistance includes physical barriers such as your skin, or chemical barriers such as the lining of your stomach, phagocytes, fever, acute inflammation, complement activation, and natural killer cells. Again, physical barriers are skin and mucous membranes, so in your nose and in your mouth as well. Chemical barriers such as your skin, saliva, and tears. And phagocytes are those white blood cells where macrophages that have now become activated to phagocytize or engulf and destroy foreign organisms. A fever can accelerate the mobilization and deployment of host inflammatory cells. Fever, because of the high temperature, will inhibit growth in those microorganisms. If you remember, heat denatures protein, and organisms are made of those proteins, right? So the endotoxin, it's also inhibited as a release from the gram-negatives, and exotoxins induce the macrophages to release the cytokines, such as interleukin-1 and tissue necrosis factor. Interleukin-1 can cause prostaglandin to be released, which can also help reset the temperature. Also, with innate response, we have the inflammatory response. So signs of acute inflammation, redness, swelling, heat, and pain. Interleukin-6 is actually released by the macrophages, which induces the liver to produce your C-reactive protein, or your CRP. Your CRP will optimizes and coats the microbe, so it's more easily phagocytized by the macrophage. If you remember, we have two pathways. We have the classical and the alternative. Your classical is triggered by an antibody binding to an antigen, and the alternative is induced by the chemicals in the cells. So basically, with our complement cascade, we have a multitude of steps, but really it breaks down to this. So the goal of complement is to lyse the foreign body, so cytolysis or lysing of the cell. Ultimately, it helps them put together enough molecules to make the MAC, or the membrane attack complex, which then punches holes in the cell wall. Anytime contents of the organism are released, they become non-viable. Optimization is the binding of the C3b to the foreign body and enhances the phagocytosis of the PMNs and your macrophages. And then we get to the generation of peptide fragments such as C3a and C5a, and that helps to enhance the inflammatory response. All right, so that was innate, and now we're going to shift gears to the adaptive immune response. This is also called specific or acquired, so you need to think specificity and memory. Antibodies are always involved with adaptive. This is not what you were born with, which is the innate. This is acquired. So we actually have two arms of the adaptive immune system. So we have the humoral, so think again, antibodies. So B cells, plasma cells, and antibodies make up the humoral, and then the cellular, which are going to be your T lymphs, your cytokines, and your macrophages. For an organism to actually cause disease, it has to have a route of entry to its host. This could be from ingestion or inhalation, from direct contact of that organism. It's going to have to have an adherence. So think of your mucous membranes. They have to have a mechanism of invasion, and then secretion or secretion of a toxic substance. So sometimes the immune response is not just to the antigen, but to the type of toxin that's produced by the organism. The type of toxins can be an exotoxin or an endotoxin. So exotoxins are proteins that are secreted by the microbe or are released during cell lysis. Enterotoxins are actually exotoxins, but they act specific on the GI tract. So an endotoxin is part of the outer membrane of a cell wall of a gram-negative bacillus. Typically, it's a lipid A, so the endotoxin cell wall of a gram-negative bacillus will release the endotoxin, and that will activate your complement and will also activate the release of your interleukin-1. Tissue necrosis factor symptoms then are going to include fever, and severe enough, it could go into shock. So if you think about toxic shock syndrome, which is typically caused by strep pyogenes or Staph aureus. We also can get a biofilm formation. So this is going to be defined as an accumulation of microorganisms embedded in a polysaccharide matrix. These pathogenic organisms use a biofilm to adhere to implants or prosthetic devices, so like a heart valve in cardiac patients or like an artificial joint in a knee or a hip. That biofilm may display reduced susceptibility to antimicrobial agents, so the organisms develop some additional resistance and will demonstrate a different gene expression, and it's able to adapt and respond to changes in the environment. The biofilm may be an accumulation of a single organism or it may contain multiple types. So polymicrobial aggregation cells in the biofilm are physiologically unique, so oftentimes they're referred to as persister cells. Approximately 65% of hospital-acquired infections are going to be associated with a biofilm. Organisms within a facility are just like their local residents. So kind of you think about it, going to the hospital, you think it's clean and sterile, but that is not the case.